So today this lecture is an informal lecture. So if you have any questions, please just let me know. So today, let's talk about your final exam. Your final exam will be a project which is using some of the tools or software you have learned in this class to do some research. First, you need to find a target, any proteins or other biomolecules you're interested. You can find it in PDB bank. First, you get the structure, and then you can use Delphi or Delphi Force or do some MD simulations and tell us some stories about your interested target. For example, if you are interested in COVID-19, you can download the structure of COVID-19 and then do some calculations and tell us what did you find in the simulation or calculations, and then give a project report, give a talk. It doesn't have to be long. It could be 5 minutes or 10 minutes. That's fine. That's your final exam. We don't have midterm. So your final exam is a project, and today I will give you 2 examples of some research on proteins using the computational biological tools. Can you see my screen now?
Yes.
Okay, I think I can't hear you. Let's see. Built-in output. Okay, so so now I will show you 2 examples of using the tools to do some research and tell a story. So for example, the first target is molecular motor. So here, for example, if I am interested in molecular motors on microtubules, so what I can do is I can first do a background check to see what's the functions of molecular motors and what's the functions of microtubules, and then download some structures, and then find maybe just 1 or 2 interesting points or interesting detailed structure or something, and do some simulations and see what happens. So for example, this is a structure of microtubule. So microtubule is like skeletons in the cell. It's connecting the center of the cell and the wall of the cell. So it's like a skeleton and also highway in the cell. So that's the overall structure of microtubule. If you look into the details, you will see microtubule is composed by tubulins. So now you're watching the movie of how the microtubule grows. So the microtubule grows from this end to the other end, and this growing direction is called its positive or plus direction. And the other end is the minus direction. And the molecular motors, they work like a delivering truck. So they deliver cargoes on the highway. The highway is the microtubule. So here shows 1 type of molecular motor, which is kinesin. The kinesin is trying hard to deliver a cargo from 1 end of the cell to another. Here, the last picture, it's another type of molecular motor, it's dynein. So the difference between kinesin and dynein is, so first, the structure of the kinesin and dynein, it's quite different from the movie and the picture. You can see the structure are quite different, and their functions are also different. Dynein moves along the microtubule from plus end to minus end, and the kinesin moves from the minus end to plus end generally. But recently, some research found some of the kinesins, they could move from plus end to minus end, working just like dyneins, but it's very rare. So this is the first difference. The second difference is for kinesins, they have another function. Besides delivering cargoes, they also involved in the cell division or mitosis. So when one cell is dividing into two cells, the kinesin is a motor which separates the microtubules and then divide one cell into two. So kinesin is very important in cell division. Therefore, there's some recent work which targets kinesin as cancer drug target, because you can imagine if you can block the function of some kinesins, so some cells cannot be divided or overgrow. So most probably, if we can kind of design some kinesins in a proper way, most probably we can inhibit the cell division, especially the tumor or cancer cell division. Then it could be a potential drug target for cancer treatment. So that's kinesin and dyneins. Because they're so interesting and so important, so let's say I want to do some research on kinesin and dynein, then what I can do is, so here let's do some background. What I can do is I can first download the structure of dynein and kinesin. You can do a search in PDB bank as shown by Wenhan. So just type dynein or kinesin in the PDB bank. You will get a lot of structures of dyneins and kinesins. So just choose a recent one with high resolution, and then you can start your research from there. Now let's see the structure of dynein. This is the structure of dynein, and this is the structure of kinesin. This is the kinesin motor. The dynein, the structure is quite different from kinesin. For kinesin, it has a stalk, which is a helix dimer, and it has a magnetic linker, and it has a motor domain, actually two motor domains, and those two motor domains move step by step and therefore work along the microtubule. So this is the structure of dynein. So for dynein, it's also working along the microtubules, but the structure is something like this. So this is a motor domain. It's like a ring, and then it has a long stalk, so it performs like a leg, and then the foot is the binding domain, which binds the microtubule. So the dynein can walk along the microtubule step by step. Okay, so when we do a research of, let's say, dynein, so there are so many things we can investigate. So one thing we were interested is what's the reason for dynein working along microtubule with a fixed step size. So every time it works with a fixed step size, and the step size is 16 nanometers from here to there. That means it's the twice of the length of the tubulin dimer, because tubulin dimer is 8 nanometers, the length of tubulin dimer. So each step of dining means the 2 of the length of the tubulin dimer. So if you do a literature search, you will see some of the researchers, they think the neck linker, which is here, it controls the step size, which can move the dining step by step accurately. Every time it's 16 nanometers. But actually it's not very convincing, because if you look at the structure of dining, the stalk or the leg is very long. So if you want to choose this part, the level or the linker to control the step size, the accuracy of the level or linker should be very accurate, because any error happened here will be amplified by the long leg, and it will cause a huge error. Even a small error happened here, it might cause a huge error. So this is the first reason we doubt the hypothesis. The second reason we doubt about the hypothesis is if that is true, if the neck linker controls the step size, then the leg of dining should be very rigid. If it's flexible, then it's impossible for this part to control the step size of the dining. So what we can do is, now you already have the structure, you have some background of the protein, and you have some ideas. The idea is we want to test if the hypothesis is true. So one thing we can do is we can first run an MD simulation. Let's see if the stalk is rigid. If you run a simulation, you will see the stalk is not rigid. It's kind of flexible, but it's not completely flexible, like moving around everywhere. It's just not as rigid as we thought. So that means it's really difficult for the linker to control the step size, which is happened at the binding domain. So there must be some other reasons or some other factors which can control the step size. So from physics, if you want to control the step size, the most effective factor should be some local interactions, like electrostatic interactions. Then to study the local interactions, we try to investigate or calculate the interactions happened here between the binding domain and the microtubule locally. So we download the structure of dining binding domain and microtubule. If you do a Delphi calculation, you will see for microtubule at the binding interface, it's red, means negative. But for the dining binding domain at the interface, it's positive. But on the other side, it's negative. So that means the microtubule or the tubulin dimer, it may attract the binding domain of the dining, and then they can bind in a proper way, like binding to the binding pocket. So this is the first finding. So the second finding, if you use Delphi to calculate the potential and use VMD to show the field lines, what you can see here is if you separate them by 20 angstroms, you will see two clusters of field lines. One cluster is between alpha tubulin and dining. The other cluster is beta tubulin and dining. So that means there are two strong forces dragging this dining binding domain to the binding pocket. And these two binding forces might be balance each other, so that when your binding domain is close to the binding pocket, those two forces will adjust the position and try to bind it to the binding pocket accurately. And then if you use Delphi force to do some calculations, what you can do is first, if we shift the dining binding domain up and then rotate it in 90 degrees, then the structure will look like that. And if you do the Delphi force calculation, you will see a lot of forces. So each arrow is a force on an individual residue. If you combine the forces on the head and also combine the forces on the tail, let's say, you will get two net forces. This is the net force on the head. This is the net force on the tail. So these two forces will form a torque. That means when the binding domain of dining is close to the microtubule or tubulin, if the orientation is wrong, then the forces will rotate it back to the correct binding orientation. So that means the torsion force will reorient the dining binding domain. Another thing we can do is we can shift the dining up and then shift it around the binding pocket above, but around the binding pocket to see if we make a circle here and we calculate the net force between the dining and microtubule at each position. What's the net force? Actually, the net force is always pointing to the binding pocket, which is a binding funnel. Means if the binding domain of dining is closed to the binding pocket, the force will drag it into the binding pocket. And there are some other tools. We're not going to introduce this tool in our class, but I can talk about it. It's just another simulation tool which can generate the binding profile. From the binding profile, we can also see there's a binding funnel between dining and the microtubule. So after those calculations, you can get the conclusion that the factors which control the step size of dining on the microtubule is the local force, which is local electrostatic interaction. So the full story of the one small step for dining should be something like this. So first, the linker has some configuration change, which rotate the ring and move the leg forward. And the local interactions between binding domain and the microtubule will adjust the forces and reorient the orientations and then attract the binding force to the binding pocket so that the dining works along the microtubule accurately in each step. Okay, so that's the story of dining. So now let's see another application, which is the application on capsid assembly of giant viruses. We're using the similar tools to investigate how does the viral capsid assemble. So this is the structure of virus. If we look at the proteins of the whole virus, so most of the virus look like that. You will see it's a spherical structure, but it's more like a Lego. So the entire structure of the entire capsid of the viral capsid, it's composed by the same pieces. So you will see a lot of pieces. It's like Lego pieces. If you put the pieces in a proper way, then you can finally assemble this format or this sphere. You will see it has some different colors. So here, different colors means different orientation. So for example, blue and green, they are the same capsid protein, however, they're in different orientation. And green and red, they're in different orientations. So different color means different orientation, but the same capsid protein. So we want to see what's the detailed mechanism of protein-protein interactions in the capsid assembly. So how the virus assemble together to form such a severe capsid. So if we zoom into the protein or the capsid protein of the virus, you will see the structure looks like that. It's a trimer, and it's a gel roll trimer. You will see it has three proteins. So each of the capsid proteins, it contains 3 chains. Each chain is a monomer, and finally they form such a shape. It's like it's a capsid, and its structure looks like a hexagon. And before our work, there are some hypotheses of viral assembly and disassembly. So some structures or some works revealed some hypotheses. They think for a virus, they first form some big patches like big triangle, big pentagon, and then the triangles, pentagons, then form into a spherical shape like this. So another hypothesis thinks that for a viral capsid, the assembly mechanism is that it starts from some tip, like here, and then it grows the entire viral capsid by adding some capsid proteins one by one. So you will see the structure of the viral capsid is getting bigger and bigger, and starting from here, and finally it grows into a spherical force, a spherical shape. So they are all from some experimental evidence. So for example here, I don't know if you can see it clearly, but it's a small piece of viral capsid. It's in a pentagon shape, which demonstrate it grows from a pentagon and then grows bigger and bigger. So in our simulation, what we can do is we can first study what's the interaction between 2 triangles, and then let's see if let's say the virus disassembles, will it disassemble inner a triangle or inter triangles? Okay, so what we can do is first we choose the boundary of 2 triangles, and then we choose 3 capsid proteins, A, B, and C. So it's a representative of most of the interactions between, actually all the interactions between each 2 triangles. In each 2 triangles, you can only have 3 types of interactions, AB, AC, BC. So all the other interactions, it's just a symmetry of those 3 types of interactions. So first we use Delphi to do the electrostatic calculation, and we see the charge distribution is something like that. So AB are the side views of a capsid. C is a top view, and D is a bottom view of the capsid protein. From the side view, you will see on some sides of the capsid, it's very negative, and on some other sides, it's positive and negative, but more positive. And on the edge of a pentagon or the capsid, it's always positive. So if you map the entire surface charge onto a pentagon, you will see the charge distribution is something like that. On the edges, it's always positive. On let's say three surfaces, it's negative, and it's a strong negative, which is shown here. But on the other three surfaces, it's generally positive, but it's not so strong because it has some positive, positive, and negative. So it's generally positive, but weakly positive. So to simplify the charge distribution of a capsular, we can use pentagons like this. Sorry, not pentagons, but hexagons like this. And we label the charges in this way. And now if you go back to the ABC that we have chosen from here, remember that ABs are in the same direction because they're blue, and C is green means they're in the different direction with A and B. So here, A, B, they're in the same direction, and C is in 60 angles difference between A and B. So if this is A, this is B, you will see the direction of A and B here is exactly the same. However, for C, it has been rotated by 60 degrees. So the interesting things happened here. So first, if you compare the interactions between A and B, you will see in A and B, so here it's positive, here is slightly positive. So here it's generate a repulsive force. But here, positive, negative, it's attractive force. And it's a strong attractive force. If you compare the interaction of B and C, you will see here is strong negative, strong positive, strong positive, strong negative. So the total interaction should be strong attractive. And here, between A and C, is positive, 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 positive. So that's repelling forces. So the entire binding force could be divided into three types: mode 1, mode 2, and mode 3. But this is not a calculation. This is just based on our schematic figure. If we do a real calculation by del phi, then you can draw the field lines. From the field line first, you see between A and B, the field line is dense here. That's a dense cluster, which means it has attractive interactions between the tip of B and the surface of A. And there's also some field lines between the surface of B and surface of A. So it's attractive force. But between B and C, you will see this attractive force is so strong because if you look at the charge distribution on this schematic figure, you will see it's strong negative, strong positive. They are matched very well. So you will see there are two clusters of field lines, means very strong attractive forces. But between A and C, as we expected, they are repelling. So there are not so many lines, field lines connecting A and C. So that's electrostatic forces calculated by del phi. Now let's see del phi force. What we can do here is, if you have, let's say, binding mode 1 means between capsular A and B, if you fix B and moving A, and then you do the force calculation, you will see the electrostatic force is always attractive. If you do a rotation of capsular B, you will see at different angles, the force becomes different. For example, if in the 30 degrees range, if you rotate it in 30 degrees range, it's always attractive. But if it's beyond 30 degrees, it becomes repulsive. So the other calculation is still using the del phi force, but if we shift the protein B or capsular B in this way, and then you will see in a certain range, there's attractive force, but if it's beyond this range, the force becomes random. So again, it demonstrates there's a binding funnel between capsomer A and B. And here, if you rotate B surrounding A, you will get similar results. So at a certain angle, it's attractive, but beyond this range of angle, it becomes repulsive. So again, it demonstrates first in mode 1, A and B are attractive no matter how far away they are. And then when they are getting closer, there's a binding funnel. No matter if you spin protein B or rotate protein B surrounding protein A, or if you shift protein B in a perpendicular way, you always see the binding funnel. And similarly, you can do the same thing for mode 1, mode 2, and mode 3. And you will see some interesting conclusions. We're not going to introduce the details here. And you can also do some magnitude analysis for the binding forces by using Delphi force, which can also demonstrate binding funnel. And by doing some MD simulations, molecular dynamic simulations, you can do some binding energy calculations and find which mode has the highest binding affinity. But in this class, we're going to learn how to do the MD simulation, but we're not going to learn how to do the binding energy calculation. That's too much for this class. But here you can at least see if you have the MD simulations, you can easily get binding energies from the simulation. Okay, so the conclusion is, okay, let's find the slides and make a conclusion. So the conclusion is in the capsid A, B, and C, actually AB means the binding mode inside a triangle, or the most dominant binding mode. For example, the big triangles. So in a triangle or in the same color, it means they're in the same direction. So that's the binding mode of A and B. So for this binding mode, it's strong. For binding mode at the boundary of two proteins, the two triangles, there are two types of binding mode. One is very strong, and the other is repulsive. So it's been shown here. So C is from another triangle. ABs are in the same triangle. So C has very strong binding affinity with B, but it's repulsive from A. But generally speaking, still, A and B, they're always binding. But if you consider the interaction between C and AB, so there are some weak interactions which might be easily break. So when a viral capsid is, let's say, disassembled, most probably it starts from the boundary of triangles. And then that's why in experiments, when a viral capsid is broken, you can observe some triangles, because the interactions between triangles, they have some weaker or repulsive binding mode, which easily breaks the triangles interactions. So by doing this, you can explain the detailed mechanisms of capsid assembly in a virus, and you can also explain some experimental findings like this experiment. Why, when the virus is disassembled, you can see some big triangle patches. Okay, so that's the second application of the computational biophysics tools on the molecular motor and then on the viral capsid assembly. So that's all for my examples. So in your final exam, what you need to do is you find an interesting topic. It could be anything that you are interested. So just find the structure and do some calculations by using either Delphi or Delphi Force or MD simulations, and tell us some stories. So what did you find in your target? Of course, you don't have to tell a big story like this. So this slides, it cost a couple of years to finish all the ideas, proposals, work, analysis, simulations. But in your case, you only have one semester. Actually, you only have half of the semester now. So it could be a small project, just use one or two tools to do the calculation. Tell us a little bit about the background of your interested target. Tell us about some basic knowledge of it. Tell us about what's your idea about your biology target, and tell us what did you find and what did you use, what's your results. That's it. 5 to 10 minutes should be fine. So each one should have one presentation at the end of the class. That's your final exam. Okay, do you have any questions, guys? If no questions, I'll see you next Monday. And on next Monday, Chitra will give some MD simulation instructions. And before that, Chitra will share some examples for the on-class hands-on lecture. Okay, so that's it for today. I'll see you on next Monday.
Okay, thank you, Dr. Lee.
Thank you. Bye-bye.
Bye.
Bye.